Yeah, so welcome everyone. This is our third seminar series for the ESEB STN Mating Systems and Dispersal. Before we start, I'm going to announce the next fourth seminar that we're going to have, which will be exactly 5 weeks from now. So this is Thursday, the 3rd of July. We currently have one speaker lined up. This is Anne Pussy from Duke University, who will talk to us about her work on primates. This will all be in an email later on as well. But yeah, Thursday, 3rd of July is our next seminar. So that will focus a bit on sort of macro species and animals, mating systems, dispersal. But today we're focusing a bit more on plants. So we have two very exciting speakers. So first up, I'd like to welcome Professor Anna Hargreaves from McGill University. She runs a group that looks at sort of the eco-evo aspects of species interactions and the dynamics of species range limits. So a perfect speaker to talk about, range limits and expansions. So Anna, I hand over to you.
Yeah, super. And you can see my slides. It's working.
Yeah.
Great. I was just saying, it's been really fun putting this talk together. So my lab, we work mostly on trying to understand species geographic distributions. We also work a lot on species interactions, and I have in the past worked a lot on dispersal. So I did some theoretical synthesis, and we did some empirical studies and an evolutionary model. And we even tried to get an experimental evolution system going, which was like a total failure. Like helped along by COVID, but also, it's hard to dabble in bacteria. But I haven't actually thought about this for several years. So I am a bit out of date on the empirical literature. So if I say things that are out of date and you know of other studies, then please tell me and especially if there's studies that you've done, because I would love to know about them. Bring me up to date. Okay, so with that caveat, I don't have to convince you that dispersal is interesting. So let me just put in a pitch for why species ranges are interesting. Species ranges are, for an individual species perspective, they are the spatial expression of a niche to a large extent, but they determine who you interact with, where your habitat is. They're really tightly related to overall population size, and so they can influence conservation status. And then, of course, they scale up once you start overlapping these distributions. They're really the building blocks of biogeography and biodiversity. So the individual processes that shape individual species ranges scale up to determine what communities and ecosystems look like where all of us live. They're also an amazing arena for looking at really fundamental questions in ecology and evolution. And I would argue there's no more interesting place to study dispersal and dispersal evolution than at the edges of species ranges. So in absolutely every case for every species, at every range edge, the range edge is set by some combination of niche constraints and dispersal. So there is always a role for dispersal. So if you take, say, a very stable, very continuous environment where I've given you a gradient where really good habitat is in green and untenable habitat is in blue, so then you would have some inflection point along the gradient where populations go from self-sustaining in here to not being able to sustain themselves here. And so this is what we sometimes call the niche limit. And if everything is, stable and eventually the species can occupy the full landscape, and you get this coincidence of range limits and niche limits. And this is kind of what, I think most of our default idea of what a range limit looks like. And if you have really good dispersal, you can even get dispersal maintaining sink populations beyond the range. So if you get continuous dispersal and the gradient's not too steep, then even in this super stable continuous environment, dispersal can influence where that range limit is. But I'm sure, if any of you are empiricists, I'm sure your study system is neither stable nor continuous. And so for most environments, there's annual fluctuation, right? So you might have, this is where the niche limit is in normal years. And then you might have some really good years where conditions are okay here, and some really bad years where conditions are not okay here. And here, dispersal really starts to play an important role. So if you have good dispersal, then your range limit can track the niche limit. So as conditions are good, you can expand. When conditions are bad, you get pushed back. But then when conditions are normal or good, you just recolonize that habitat. But if you have bad dispersal, then you get pushed back in bad years and you never regain that ground in good years. And so if you have bad dispersal, what you can find is that the range is just constantly, in most years, dispersal limited. And so again, now you have dispersal playing a much bigger role in the location of this range limit. And then, of course, I think what everyone thinks about all the time now is not just a fluctuating environment, but a directionally changing environment. And we think about this because of climate change, and in theory, at least at the sort of cool limits of species ranges, if temperature is controlling the range limit, which is, we've done a lot of work on that in my lab, and it's definitely not only temperature usually, but to the extent that warming improves the environment, then you can get these range expansions. And so now dispersal again plays this huge role. If you have good dispersal, you can track your climate niche, and if you don't, you can't. So we have this role for temporal variation, but of course, in every landscape at all kinds of spatial scales, there is also, sorry, this is just the visual depiction of what I just said. You have spatial variation as well. And so landscapes are patchy at the macro scale. They're often patchy at the micro scale. And there's lots of theory and not as much empirical work that suggests that range limits can also overlay gradients in the amount of habitat. So in this very extreme case, you might have no gradient at all in the quality of these patches. All of these patches are awesome, but they get smaller and farther apart. And when this happens, what you might have is, the species occupies areas where the populations are big enough to be self-sustaining all the time. And then you have dispersal sort of colonizing these smaller and further away patches. And the smaller the patches, the smaller your population is, the more likely you are to undergo just random extinction. And so you can get what are called metapopulation dynamics at range edges, where the range limit occurs wherever extinction outpaces recolonization. So you get these periodic winking out of populations. If you have good dispersal, you can recolonize those patches. If you have bad dispersal, you can't. And then you have, little patches that are either very small or far away that just never become part of the range. And so one of the kind of distinguishing features of metapopulation dynamics is A, dispersal is super important. B, you expect to have high quality habitat that's unoccupied beyond the range. And then, of course, you can get gradients in both the amount and quality of habitat. And really all that happens here is that more of these patches become unoccupied. So that was like a whirlwind overview of dispersal theory for species ranges. But I think you can see that like in every single one of these scenarios, your relative dispersal ability is key in determining where that range limit is. If you have low dispersal, then you often end up having suitable habitat that's unoccupied. If you have high dispersal, then you can track your range in space and time, and you may even get these sink populations. And so we did a meta-analysis of experiments that had moved species beyond their current range. And this is a classic test of dispersal limitation. So if the species does okay beyond its range, then you infer that its range is dispersal limited, at least in one way, one of those many ways we just talked about. And we found that 26% of range edges were limited by dispersal. And this was mostly for latitudinal gradients. Almost as many range edges were extended by dispersal. where the actual edge population didn't seem to be viable itself. And these were mostly elevational range edges. And so you can see that it's not just the species' innate dispersal ability, it's also how steep the gradient is. So along an elevational gradient, you don't have to disperse very far to disperse along sort of an equivalent climactic gradient, whereas for latitude, you have a much higher dispersal demand to get to a new climate. So, in about half of the species that we looked at, dispersal is really modifying the range limit. So that's the ecology of dispersal. But of course, I mean, for this group, I assume you're also all thinking about dispersal evolution. And so dispersal is a labile trait. It evolves. It can evolve really rapidly. And the processes that select for and against dispersal can be really pronounced at range edges. So again, this is a pitch for why range edges are particularly interesting from a dispersal evolution perspective. So one of the big things that selects on dispersal is spatial variation in habitat. So once you have spatial variation, then you risk dispersing to bad habitat. So you can imagine that if you live on this little island, dispersal is risky because there's a really good chance that you'll land in this blue area where, essentially your seeds die. And so generally, the more spatial variation you have, it selects against dispersal. And you get reduced dispersal. And so it's possible that if you have range limits that are characterized by increasing patchiness, you actually would have dispersal evolve downwards at range edges. And of course, this has implications for, range shifts and how well species can then track fluctuations at their range edge. Another thing in the dispersal evolution literature that selects really strongly on dispersal, but in the opposite direction, is temporal variation. And so if you have habitat that is fluctuating in quality, especially if you have habitat that goes from viable to totally unviable, then if you don't disperse, you risk eventually going extinct when your little patch of habitat becomes unviable at some point. And so temporal variation generally selects for dispersal. And to the extent that temporal variation is more important at range edges, we have the opposite prediction then. So you might expect increased dispersal ability at range edges. There's other things that select. I don't know how many of you are like dispersal evolution nerds, but in the kind of world of dispersal evolution theory, there's other big selective forces. You can escape competition. Kin selection is this like very weird thing that comes up in models all the time. But also trait correlations, right? So a lot of dispersal, it's not like it's not like coat color, right? Dispersal is this composite of many traits, and those traits can be under selection for totally different reasons. So you can have dispersal evolving up and down due to selection on things other than dispersal. So we have these, at least two reasons why we have completely opposite predictions for what dispersal might look like at species range edges. What does the empirical evidence say? Well, there's loads of evolutionary models. Like dispersal has been really well studied by really creative people doing interesting things. And, that's where all of that theory comes from. There's very little data. So it's exciting to hear that you guys are going to be brave enough to try and collect it. Dispersal is really hard to quantify. I mean, on mammals, you can track them sometimes through collars, but like that's hard. That's not easy. Your sample sizes are low. For plants, it's particularly annoying because seeds are really small and pretty much impossible to track. So we don't have very good data. There's been very few attempts to measure variation in dispersal towards range limits. Like, we found very few when we analyzed the data about 10 years ago. A more recent review found only 5 studies, of which only 2 of them were on native species. So, so I would argue, like, here's my pitch. If you're going to collect dispersal data, think about where those data come from, because, we have like okay data on dispersal traits per species. And yet we know there's incredible variation within species. And to my mind, that is by far the most interesting part. That's where all the really interesting questions are coming from. But there's a huge need. So if that's something this group's interested in, that would be amazing. Okay, so that was sort of a synthesis of, normal range limits. A lot of effort thinking about dispersal has focused on expanding range limits. And of course, there's two reasons for this. One is that you can't get a range expansion without dispersal. So dispersal is a necessary first step to range expansion. It has to happen. There's other factors that have to happen afterwards, but like if you don't have dispersal, none of those matter. So it's this key trait that's involved in range expansion. And once you're talking about a range expansion, you have also these additional factors that come into play that influence dispersal. So first of all, escaping competition becomes more prevalent. So if you're going from where your species exists to where it doesn't, then now you completely escape intra-specific competition. There's a colonization advantage often to being the first genotype to arrive in new habitat. So for example, if there's priority effects, you can sort of increase your genotype's frequency in the population just by getting there first. You have first access to all the cool new resources. There's also this process called spatial sorting, which is really only articulated relatively recently. And the very brief summary is that essentially, if you have a range edge and then you have some genotypes, the high dispersal genotypes disperse beyond it. They don't have anybody to mate with except each other. And so you have the high dispersal genotypes mate with each other, and then they send propagules off. And again, those are the highest dispersal genotypes. They mate with each other. And so you have this sort of evolution that is really just about the spatial sorting and assortative mating of dispersal genotypes, which really only happens in range expansion. So there's all this theory that suggests that dispersal evolution at expanding edges might be strong and then accelerate the rate of expansion in ways that are really cool and exciting. Does this actually happen? There's great evidence from invasions. So an invasion, what you have is you have expansion into uniformly good habitat, right? So you haven't reached your own niche limit yet. All the habitat is great. And it's often relatively free of competition. So not only are you escaping intra-specific competition, but often in models or in experimental systems, there's like nobody else there. And then in real invasions, there might not be an equivalent of you there. So for example, this is a beautiful study on Arabidopsis where they just set up these trays and they let the plants disperse seeds, and they had this like rotating landscape. This work I think was done in Switzerland actually by Jen Williams, Jonathan Levine. And so they had this ongoing expansion, and they showed that the high dispersal clones increased in frequency. There's been groups studying both bean beetles and flower beetles that create these like awesome landscapes, again, where the beetles are allowed to disperse. And they've shown also rapid evolution of increased dispersal during these expansions. And there's much less evidence from the field, but there's beautiful work done on cane toads in Australia, where a lot of the field data that you have is space for time substitutions. So they'll compare a population at the invading edge versus more centrally, but that's not perfect. But if you, the cane toad data, they have populations followed through time. So they have really like the kind of data that you want that shows that it is an increase in dispersal ability. And essentially cane toads, what happens is that they're more curious at the expanding front, so they change places more often, and they have longer, better dispersing legs. And so you have this accelerating invasion front. And what's interesting is that the dispersal goes up and then it goes back down. So the high dispersal genotypes are really at the range front. Anyway, so there's this beautiful evidence from invasions. However, mostly what we're talking about, or at least what I think about, are range shifts. And it's much less clear for range shifts what's going to happen. So in range shifts, you're not expanding into uniformly good habitat. Presumably some niche constraint imposed your range limit before, and it's only getting a little bit better. So you can disperse into new habitat, but in theory, it's worse habitat than you already occur in. So you're dispersing against this environmental gradient. And of course, with a range shift, it's already occupied, not by your species, but you're not going to be the only toad in Australia if you're undergoing a range shift. And so you have reduced colonization advantage in marginal habitat. There's less competition escape. It's also possible to disperse too far and overshoot the niche limit. So the selection is much dampened. We argued this sort of conceptually, but there's been a recent model by John Benning and colleagues that show that indeed, if you put environmental gradients into an invasion scenario, the upward evolution of dispersal is really reduced. And in fact, you can get the downward evolution of dispersal from these environmental gradients if they're steep enough. But again, as far as I know, there's almost no data from the field for range expansions, for range shifts. Okay, so will dispersal evolution facilitate plant range shifts? I think about this a lot. I am a little bit skeptical. Again, the selection for expansion to marginal occupied habitat is much less. 30 to 60% of plants have no apparent adaptations to facilitate dispersal. And so, this is something that's happened in my lab is we're like, yeah, dispersal. And then, you pick a study species for a series of reasons and it has gravity dispersed seeds and they go 2 meters and there's no dispersal trait to measure. There's no dispersal trait really to select on. Like the best you can do is height, which of course is correlated with like a million other things. So it's not totally clear to me like how the majority of plants would evolve greater dispersal, even if they wanted to. And then finally, another thing that's really frustrating is that really it's long distance seed dispersal that matters. Like that's how you get a range expansion. It's not by dropping your seed like a meter away. It's by chance that seed gets picked up somehow and moved like 200 meters away. But long distance seed dispersal is almost impossible to quantify, although I'm starting a project this summer that we're going to do our best. It's often stochastic, so it's quite hard to model. And it often doesn't use normal dispersal traits. And so it's hard to add long distance dispersal onto the end of the dispersal kernel tail. And that doesn't mean that it won't evolve. It just means it's really hard to study. Okay, so fine, it's hard to study, but imagine if plants figured this out. Would dispersal evolution facilitate range shifts? Like, could it be the savior that we're all looking for? And here I propose to you a thought experiment. So imagine that every single species at its expanding range edge evolves some magic trait that enables it to perfectly track climate change. So now you have no dispersal legs. Everybody's tracking their climate niche. Are we saved? I would argue that no, we are not saved, because what you have is the genotypes that were at the leading edge expanding and tracking climate. But what has happened to all of the biodiversity in the populations that have gone extinct? So if this is your range and climate kind of shifts it like this, these ones track climate by dispersal, but the contracting edge tracks climate by dying. And so just being able to track climate, like we think so much about this, but I think we're thinking about it wrong. Just being able to track climate, in like this world that I've proposed to you, this thought experiment gives you one genotype, or like this incredibly depauperate new world of species that are where they should be, but you've lost a ton of genetic diversity, and the next fungal pathogen that comes along is just going to take them out. So I have thought a lot recently, forever, about what is going on at contracting edges. because dispersal at these contracting edges also matters. If those genotypes can't track climate or move with the range, then we get these huge erosion of biodiversity. And also the contracting edge, in theory, is where all of your heat adapted genotypes are, right? So like those are the last genotypes that you want to lose in a warmer world. We know almost nothing about what's going on at contracting edges. That goes for dispersal, but that also just goes, they're modeled way less often than expanding edges. Expanding edges are relatively easy to model. They're fun. Contracting edges are annoying to model. They're not fun, and we don't do it. And so, it's not just that we don't have empirical data, but like we have almost no theory on what should happen at contracting edges. And so here it's useful to think about, okay, well, how do range edges contract? I mean, I think what we think about is you just get this environmental deterioration. And so these populations die out. And that's kind of what I was thinking about in my thought experiment. If you get the deterioration of an environment, these genotypes, if they're plants, are going to be suffering. They're going to produce fewer and fewer seeds. A major component of dispersal is actually just how many dispersers you send out into the environment. And so if anything, you might expect dispersal to be reduced at contracting margins, not necessarily evolutionarily, but ecologically. If you have, the other way it could happen is if you have this patchy environment, you could get the winking out of habitat patches. If this happens, what you have is an increase in spatial variability where now it's super risky to disperse if you're this genotype, right? Your chance of making it to another habitat patch are really low. So in patchy environments, if you get the winking out of patches, you actually might expect dispersal to evolve downward at these contracting range edges, creating what's essentially an evolutionary trap, where now they're even less likely to move with climate change than they were before. So back in my one and only attempt to do a dispersal evolution model, we thought about this, and our question was, will dispersal ever evolve upward at contracting edges? And I should have animated this. You don't have to pay attention to that now. But essentially, when we think about a range following climate change, we often think about the species having a single climate niche. So that at the expanding edge, these populations moving, that's great. And at the contracting edge, these populations just die out. But you can get local adaptation to temperature within species ranges. If you have really strong, imagine you had extremely strong local adaptation, then each of those little populations has its own climate niche, and each of them will be kind of evolutionarily motivated to track that niche through time. And so we thought, well, okay, if you had strong local adaptation, could you actually get selection across the range for increased dispersal in the way that you get it at the range edge? So we created this model. This is the range. This is time. And so we populate it here. And then for a long time, nothing happens. The climate is stable. The rainbow colors show you the dispersal genotype. Blue is low dispersal. So here, everything's fine. It's boring. There's not really that much need to disperse. Some plants do it, others don't. As of this dot, we start shifting the climate. So this is kind of a very simple climate change scenario. And what you see, this is the leading edge. So you see the range move through space with time. And you see the evolution of high dispersal genotypes at the range front. So this is exactly what you expect, but all the habitat is good. Climate is the only thing that's controlling the habitat. So classic invasion scenario. If you make the habitat at range edges marginal, then you get the same thing, but because now this habitat is not as good, the range lags climate change. So you don't get as strong an evolution, you don't get as good range tracking. If you add local adaptation, and you can see, sorry, the other thing I meant to point out is that you don't really get dispersal evolution at the contracting edge. you get this smear back from the leading edge that's a bit helpful, but there's not really any evolution happening here. If you add local adaptation, you start now to get dispersal evolution throughout the range, which is what we predicted. So that is kind of exciting, except what you also have is much more extinction, because now everybody is constrained in where they go. Everybody, has less habitat to work with. And the increase in dispersal evolution across the range from local adaptation was not enough to save this species. And overall, with local adaptation, although it helped dispersal evolution, you had more simulations where the whole thing just went extinct. So that's not very inspiring. The kind of key points that I want to leave you with, first of all, I think it's awesome. There's a whole group of people interested in dispersal. Again, I think that species range edges are such an interesting place to study dispersal. It's the key determinant of where range edges occur and how they move. It can evolve quickly at expanding edges, but it's not clear how this applies to range shifts, and it's probably not as optimistic a scenario as you would get if you're just thinking about invasion. And again, dispersal and just evolution in general at contracting edges, I think is hugely important and really understudied. So if you ever want to get in touch, that's my email address. This QR code will take you to a folder of the papers that I talked about during the talk. And if we have time for questions, then that's awesome.
Thanks very much, Anna. This was super, super interesting. If people want to ask questions, please feel free to just unmute yourself or put up your hand.
I can maybe, whoops, sorry, Matt, you go.
No, go ahead. Go ahead, Greta. I'll ask after you.
Okay, I was really curious. You mentioned that you have a project starting where you will try to measure long distance dispersal. I was just curious how you go about it.
Yeah, okay, that's fun. This is like my new favorite project to talk about. We go about it through collecting poo. And so this is a collaboration with Jake Alexander, who's at ETH Zurich. And his lab ran a pilot study in the Swiss Alps, and now we're doing it in Alberta where we work and the Swiss Alps. And so the idea is along an elevational gradient, you can survey where species occur. And then you can wander around and collect like poo from ungulates. There's livestock in some places, and you can take that poo back to the lab and germinate it and see what seeds you had. And then you can actually look at how far above species ranges plants are being dispersed already. So, if you only find a plant up to 600 meters, but you can germinate it out of poo that you collected at 800 meters, then that's obvious evidence that the species is being dispersed at least 200 meters beyond its current range limit. So it's this really cool way of quantifying. I mean, even that may not catch the really long distance end of dispersal kernels, but very few seeds have any mechanisms that facilitate being eaten by ungulates, for example. It just kind of happens randomly, and probably lots of them get crunched up, but a surprising number of seeds make it through. And so then you can kind of get the data that way. And then in theory, you can combine that with movement data from like movement ecologists to try and kind of back of the envelope calculate, if species are moving these distances, you can get like a distribution of movement frequencies. You, in theory, could estimate a long distance dispersal tail for seeds that way.
Yeah, that's very clever to use animal dispersed plant.
Yeah. I mean, and it's fun because, at first when we were talking about it, I was like, none of my plants get eaten by animals. But then you look at the list of Swiss plants that do, and you're like, oh yeah, probably lots of plants get eaten by animals. So yeah, it's fun. We're going to do that this summer.
Amazing. Good luck.
Thanks.
My question, I don't know if it's a fully formed question or just I might just ask you to pontificate on something. So you're thinking about what are dispersal traits in plants where everything's for many plants which are just gravity dispersed.
Mhm.
And I was thinking about something, I don't know, maybe you thought about this, but like timing of dispersal and how long a plant hangs on to versus lets go of seeds. And if that could be a dispersal trait, and that might interact with like environmental parameters that do the dispersal. So I was wondering if you thought
Yeah.
about, like timing.
Yeah, that's an interesting idea. So like there is cool work for asters that show that the different seeds have different attachment strengths to the plant. And so what that means is that some seeds get dislodged really easily and they get picked up by like gentle breezes and blown a few, meters. But some seeds can only get dislodged by big gusts of wind, which then in theory should carry the seed much further. And so this like difference in attachment strength, I mean, you could think of it probably not like a bet hedging strategy, but a strategy to generate variation in dispersal distance. And so your idea about timing would be similar. So if you spread your seed dispersal out over a longer period, you have a higher chance of catching those stochastic events, like the one deer that comes along and eats you, or the species that we work with a lot. It doesn't really have any dispersal traits, but if your hands are wet and you're trying to count the seeds, they stick to you. And there's not any mucilage, there's not any adaptation. It's just like wet things are sticky. So, those seeds, if they happened to stick to a deer walking by, could go really far. So, yeah, it's an interesting idea. I mean, this is, I think the trick would be showing that it was a dispersal trait as opposed to like, plants flower over a long period of time, which could be about pollination. It could just be about like, constraints about how you grow. So you could probably empirically demonstrate that it was good for dispersal, but it would be a real challenge to show that it was for dispersal. But it is a really interesting, it's an interesting trait. And I mean, I think, like that Arabidopsis study, it was the only dispersal, like Arabidopsis has like mustard seeds, right? Like they definitely don't have any dispersal mechanism. And it was really just about how tall the plant grew. And so, it's one of the tricky things with dispersal is that it's a really small fraction of species that has a cool dispersal trait that you can work with. And lots of them, you have these kind of like nebulous composite traits. But yeah, no, timing's an interesting idea. I hadn't thought of that one.
Well, I wonder if like at the individual level, if there's like a higher variance in seed retention increases the likelihood of dispersing via some means. But
Yeah.
if you drop all your seeds at once, that's like a risky strategy.
Yeah.
So, but I
Yeah.
see it
No,
as like a tough
totally.
trait because it's also like dehiscence of a fruit is so dependent on the temperature and the humidity. It's like
And
it's
how
tough.
old the fruit is. Yeah. Yeah, totally. But yeah. I mean, it definitely like conceptually makes sense for sure. Yeah.
Okay, I might have a question if no one wants to go. Yeah. Thank you for the talk. I really like those kind of topics, so I really enjoyed. Thank you. Just a vague, I mean, my question is also not very clear, but like I'm kind of very intrigued about all these topics on spatial sorting
Mhm.
because you can really see this like as a population structure somehow where some kind of like a population would just move along together.
Mhm.
And one can imagine that then there should be some kind of genetic consequence in the time somehow, like
Mhm.
some kind of inbreeding and therefore also, I don't know, reduced diversity to kind of adapt.
Mhm.
And I don't know if I'm not sure how much, I mean, I know the paper you mentioned, but I don't know how much of this is actually known uh, topic. And
Yeah.
if you have any thought actually on like how actually viable all this spatial sorting is also. I don't know.
Yeah.
Yeah.
Okay. I think there's three things I want to say. Let's see if I remember all three of them by the time I'm done. I think there have been good models on spatial sorting, like including that original paper that show that theoretically it's possible. In lots of ways, I mean, I think of a spatial assorting as like functionally just assortative mating, but kind of enforced assortative mating. But in lots of ways, it's pretty conceptually similar to just successive founder effects, which whenever you get sort of like a pulled range expansion where you have a genotype, it gets there first, it colonizes, it sends out the next before all the other genotypes catch up. Then you have these, new populations being founded by not very many genotypes. And then of those not very many genotypes, they send out some to found the next one. And you get an overall loss of genetic diversity. There was a paper actually that I totally meant to include in this talk that I forgot about, but it is in that QR code folder, which was led by Megan Bontrager, who's at the University of Toronto now. And we did a meta-analysis of, it's like quite complicated, but essentially studies that looked at local adaptation and compared. What we were interested in is like, is there a difference in the signature of local adaptations across species ranges? And what she found is that there is often local adaptation to extreme temperatures, but there's often just generally low quality genotypes at cool range edges. And so because our data set is mostly temperate, like Europe and North America, and those cool range edges are often post-glacial expansions. And so what we think is that this is a legacy of the range expansion. So because you have the successive founder effects, I mean, there's all kinds of theory. You can get what's called gene surfing, where you can get actually like just bad genes sweeping through range edge populations. So some combination of founder effects, loss of diversity, maybe gene surfing, lead to a deterioration in quality, population quality at cool range edges. So the paper's like very dense in data, but yeah, it's, and we wouldn't be able to sort out like the influence of spatial sorting on that. But empirically, we see that these kind of post-glacial expansion range edges are often kind of crappy populations. And then from the modeling world, I think that has been shown as well. But honestly, like Greta probably knows more about the modeling current state of affairs than I do. But I think, so it's one of those things where you can't like tie them together, but both branches of evidence suggest that, yeah, when you get that range expansion that happens by these kind of leaves, then that is overall bad for diversity, and that can last a long time. Like the post-glacial expansions, those are at least 5,000 years old, and the populations have not recovered that genetic diversity. Yeah, great question.
But I will look at the paper. Thank you so much.
Yeah, you're welcome.
So I think in the interest of time, I think we'll have to move on, but I'm sure if you have any more questions, feel free to send emails to our speakers. So thanks again, Anna, for your very interesting talk. And now I'd like to introduce our second speaker. This is Professor Matthew Koski from Clemson University in the US. He runs a group that looks at the ecological and evolutionary processes that are generating diversity in flowering plants. And we're very interested to hear what you have to speak about for our STN today. Thanks, man.
All right. Thanks, Ella, for the introduction, and thanks to the organizers of this. I'm really excited to get to share some of my work. Can you all see the screen or the slides?
Yeah.
Yes.
Okay, cool. Um, so like Anna, I haven't actually ever given a talk on this project. A postdoc who's here, Anita Cisternas Fuentes, kind of headed up a lot of this work, and she has talked about this, but I haven't. So I was excited. It was exciting to get a chance to spend some time with it. And I think, yeah, like there was some good introduction into the work that I'm going to be talking about from Anna, but my focus is less on dispersal and more on evolution of reproductive systems. So I'm going to be talking about legacies of historical colonization on population genetic structure and reproductive mode. And so during the Anthropocene, with warming climates, ranges are on the move. So we know that a common response to warming is upward elevational shifts and poleward latitudinal shifts. And we can see that in a number of systems, especially plants. So in this figure on the bottom right, this is the elevational distribution of a woody and a non-woody species, historically 1905 to 1985 in the dotted line, and then more contemporarily. And we see these rapid upward elevational shifts of climate tracking. And these range margin populations, especially those at the leading range edge or the cold front, are critically important for determining responses to continued climate change. They're likely to be the ones dispersing into the new available habitat with climate change. So these contemporary range shifts that we're seeing are not new per se. So past range shifts, like Anna was touching on, or Anna was touching on with glacial retreat and expansion, we can use those to look at the legacies of past historical range shifts to think about the consequences of more contemporary range shifts. So this is a classic example of looking at oak pollen over time. And on the farthest, my left, this is the Laurentide ice sheet during the Pleistocene glacial maximum in blue. And oak pollen was really restricted down to glacial refugial populations in the southeast of the United States. And then as glaciers receded over time, populations expanded northward, and what we get is the establishment of more leading range edges and trailing or rear range edges. So these historical range expansions set up some predictions with a lot of good empirical data about how historical expansion and range filling has structured genetic diversity of populations. So this is work from Hampton Petit from 2005, so a bit dated, but a lot of these predictions hold with additional data. But this is a cartoon showing a rear edge or a contracting range edge and a leading range edge. And you can see these kind of more patchy habitats at both range margins. And due to serial founding events from the rear to the leading range edge, we have predictions about how leading edge populations should have lower genetic diversity and reduced gene flow among populations. So they should have experienced a lot of drift and founder effects during dispersal towards the range edge. This is not necessarily a great thing when we're thinking about these populations at the leading edge being kind of the future of the species, because this reduced genetic diversity and reduced gene flow, it could hamper adaptive responses to environmental change. So it's really important to understand the genetics across the range, especially at the leading range edge. So while many traits, or really any trait, can evolve with range expansion due to founder effects and drift, reproductive traits are ones that we know very well to be associated with dispersal. And so these serial founder events towards a range edge can structure reproductive modes geographically, and this invokes the idea of Baker's law. John Panel, I think, is part of this group. I don't know if they're here right now, but they've done a lot of work on this kind of theoretically and kind of asking whether it's truly a law, because a lot of systems don't actually follow it. But the idea is that following long distance dispersal from a source population, uniparental modes of reproduction should be favored by selection. And that's, in this case, I'm showing black dots as self-fertile, gray dots as self-incompatible, and showing that if they both make a long distance dispersal event, the self-fertile individuals are going to fare better. That's because at these new populations, they might be mate limited, so few individuals to mate with. And in the case of many plants that require pollinators, there may be pollinator limited as well. And so there's good evidence for uniparental sexual reproduction evolving at range edges and with range expansions. And just one example was what I worked on for my postdoc. We worked on this flowering plant that's really widespread called Campanula americana. It has the ability to self-fertilize, but it does a lot of outcrossing as well. So it's a mixed mating species. And we grew up plants from several populations across its entire latitudinal range. And we scored the ability to autonomously set fruit without pollinators. And so each dot's the autonomous fruit set. And then on the X, what we did is we used population genomics approaches to pinpoint the most likely origin or the glacial refugium for this species. And so this is the distance from the glacial refugium. So you can see the x-axis as being the trailing to the leading range edge. And there's a really beautiful pattern here that populations at the leading range edge have evolved at least the ability to more readily set fruit without other mates. So there's also good examples of this in island colonization as well, and several other systems. So Baker's law has really been studied more in terms of sexual uniparental production, and that is self-fertilization, but it should apply to clonality as well, because as a mode of uniparental reproduction. So it's less studied, though, in the context of clonality, but the same applies, that if you have a highly clonal and less clonal genotypes in source population, following long-distance founder events, the highly clonal individuals should fare better if there's mate limitation. So more recently, I found this really awesome example of this in actually a large macro sea algae. And so this is a northern Atlantic species, and it's basically started to inhabit the Baltic more recently with glacial recession. So it's moved from the Atlantic into the Baltic and northward into the Baltic. And this study was looking at population genetic structure of the algae. And on this x-axis, this is population identity organized from the left, more trailing, to the right, more leading range edge. And this is showing inbreeding coefficients. Strongly negative inbreeding coefficients were found at the more leading range edge. And really negative FIS indicates really high clonality in plants and many other organisms. So this is evidence that these more leading range edge populations tend to be really highly clonal. And so there's some evidence that this is happening, or Baker's law is happening with clonality. So plants are interesting in that they can do uniparental reproduction through both selfing and vegetative propagation. And so there's sexual reproduction and asexual reproduction in plants, and most plants use both mechanisms. And so in the realm of sexual reproduction, we have primary selfing species, mixed mating species that are facultatively either selfing or outcrossing, and then more obligate outcrossing, about 40% of plants. And these are obligate outcrossing simply because they have self-incompatibility mechanisms. So there are genetic mechanisms that bar self-reproduction. So really the mixed maters and the primary selfers can employ uniparental reproduction via sexuality. A lot of plants also are vegetatively clonal. So there's an estimate of about 80% of plants having vegetative clonality. And then there's also parthenogenesis, which I'm not going to be talking about today. So what's interesting is that there are kind of trade-offs between uniparental reproduction on the sexual and asexual side in plants as well. So for example, there's this really great system, Solanum, the tomatoes, and Solanum has species that are self-incompatible and species that are self-compatible. And 100% of the self-incompatible species are vegetatively clonal, whereas only 31% of the self-compatible species are clonal. And so this suggests some sort of trade-off in uniparental reproduction mode. And so I'm kind of paraphrasing, but Vallejo Marin and O'Brien suggested that clonality or vegetative clonality might relieve selective pressures that favor the breakdown of self-incompatibility. So it's basically like, why break down self-incompatibility mechanisms when you can uniparentally reproduce vegetatively? But what this also suggests is that there are many species that are vegetatively clonal are also self-incompatible. And I think that presents a bit of an issue, especially when thinking about uniparental production at our leading range edge. So what is the fate of these highly clonal, but also self-incompatible taxa at leading range edges? So going back to this idea of long distance dispersal and clonality, if clonality is favored following long distance dispersal, you get really strong mate limitation. And so sex could be lost altogether. And so if a population is just highly clonal and it's lost sex, you're not getting recombination, you're not getting new genotypes. And that might be really detrimental for adapting to usually stochastic environmental conditions at range edges. So this selection favoring clonality and reduced sexuality could lead to the extirpation of these populations. So remember this because this is the most dire kind of scenario, but I'm going to come back to this later and say maybe it's not all that bad. So been working on these ideas of kind of the balance between sexual and asexual reproduction with range expansion in this plant called silverweed cinquefoil or Argentina anserina. It was historically Potentilla anserina for plant people, so I made kind of switch between Argentina and Potentilla. It's a really awesome system for studying range expansion, clonality, sexual reproduction, because it has this global distribution. So in this map down here in blue, I've just kind of highlighted the habitat that this species inhabits, and it's really widespread in the northern hemisphere and then also in temperate regions in the southern hemisphere. It has self-incompatibility. So it requires pollinator vectors to transfer pollen between non-related individuals. And then it's highly vegetatively clonal. And this is just kind of a cartoon down here of how this plant does vegetative clonality. A maternal plant produces these above ground runners or stolons. And along this stolon, it produces plantlets that are genetically identical to the maternal plant, and they can become reproductive sexually immediately in the same season. And this stolon degrades after the first or after the season, and this plant becomes free living, and so does this one. So you can get really kind of rapid clonal spread in this species. So we work on this system along an elevation gradient in the southwestern Colorado and the southern Rockies. And so this plant spans really wide elevational gradients throughout the West. And we work on a gradient of about 1,000 meters down in the San Juan Mountains. And so if you're familiar with this part of Colorado, this is Gunnison County, and Rocky Mountain Biological Lab is up here. We don't work at Rocky Mountain because all of our sites are much further south. And so this is a digital elevation model of our region of interest. And at lower elevations, our populations are in the Gunnison River Valley. And then we go south mostly to the high elevation plateaus of the San Juan Mountains. And these low versus high elevation populations are ecologically really different. So this is some of the crew in the field, and Anita pictured left here. I'm going to be talking a lot about her work. But at low elevation, this plant inhabits these wet meadows, grassy meadows, in a matrix of really dry sagebrush steppe habitat. So this plant is really tied to wet areas. And I call these low elevation because we're still on the Colorado Plateau, so they're actually still quite high elevation. At high elevation, these populations occur at the edges of glacial kettle ponds. And so these are basically depressions caused by glaciers that have melted. And Argentina anserina grows on the edges of these ponds, but also along the edges of alpine streams. And so we're at about 3,400 meters in these populations. So thinking about historical glaciations, we often think about the massive ice sheets that, like the Laurentide and the Cordilleran in North America, that covered most of Canada and parts of the northern US. But during the Pleistocene glacial maximum, high elevation sites in the west were also under glaciers. And so this is just a map. Everything in white basically was glaciated during the last glacial maximum. And so we are working down here in Southwest Colorado, and the San Juan Mountains are a high elevation range that's pretty rugged, and they were under a massive glacier that, and deglaciation lasted about 7,200 years and was completed during the late Pleistocene. So our populations at high elevation plateaus at the San Juans are likely more recently colonized than lower elevation populations. So we wanted to just confirm that. We wanted to ask whether high elevation populations exhibit the ecological and genetic signatures of leading range edges. So from low to high elevation, we would expect that there is a decline in population density on the landscape if these high elevation sites are more recently colonized and are few or far between. And then we'd expect that they have a decline in effective population size and are increased in isolation. So going off those kind of classic leading range edge ideas from Hamp and Petit. And so how we went about this is we just to test whether population density on the landscape differed across the gradient, we used a collections-based approach. So I took this digital elevation model and I calculated the area of land in 100-meter elevation bands. And then we looked at all herbarium records for Argentina anserina. And then because there might be collector bias in where people are collecting plants, for example, at high elevation, there might be fewer collections. So we enumerated all non-focal plant accessions within our bounding box, and we relativized Argentina anserina by all plant collections. So we have a standardized estimate of how dense populations are on the landscape with elevation. And so this is the data. So across our elevation gradient, population density of our focal species declines really dramatically on the landscape. So what we're seeing kind of here is our lower elevation populations might be this more continuous range, and our high elevation populations are more patchy, fewer and further between, representative of a low leading range edge. Coupled with population genomics data, I think there's stronger support. So in 13 populations spanning the elevation gradient, we used DD-RAD population genomics approaches to build a population level phylogeny. And with that phylogeny, we estimated population divergence time as its node age. We don't have a really well-dated population level phylogeny, but if we assume the substitution rate is proportional to time, we can get an estimate of when these populations were colonized. And then we looked at within and between population genetic diversity metrics using Genelex and any estimator. So this is the population level phylogeny for our focal populations. The phylogeny here is a maximum likelihood phylogeny with bootstrap support at the nodes. And the color and the number associated with each population is meters above sea level. And it's related to the color on this digital elevation model. What I'd like you to see is that down here, closer to the node, we have lower elevation colors. And then up here in the phylogeny, the higher elevation green populations are clustering together further away from the node. And if we look at an estimate of divergence time as a function of elevation, these higher elevation populations seem to have been colonized more recently than lower elevation sites. We also see a longitudinal effect of divergence time. And we are on the western slope of the Colorado Rockies. So the water flow is going from east to west. And this species is tightly associated with water. We don't know a ton about dispersal, but there is evidence that the seeds do float. And so there's the potential that this may be water dispersed. And so it makes sense that we would get patterns of kind of newer founding from west or in the western portion of the range because the water is flowing in that direction. But the big point I want to get across is that higher elevation populations appear to be more recently colonized. We looked at between population genetic diversity, and the high elevation populations have stronger FST, meaning they're more isolated from one another, whereas the lower elevation populations are more closely related to one another. And then the kind of kicker is that we see this really rapid decline in genetic effective population size. So our high elevation sites are characterized by high isolation, low genetic effective population size, really strong patterns of leading range edges. So when we were looking at within population estimates of genetic diversity, we looked at FIS. And what's interesting is that all of our populations have FIS lower than 0, and that's because they are self-incompatible. So they can't inbreed at all, so we don't get high levels of inbreeding. What's really interesting, though, is that there's really strongly negative FIS, especially at higher elevations. And like in that algae example I was talking about, high FIS is indicative of high clonality. So this is genetic evidence of potentially high clonality at high elevation. So what we wanted to do is kind of look at, are these populations at high elevation simply more clonal because when they get to these edges of glacial kettle ponds, there's less competition, so they can simply spread out and be more clonal. So there's like more ecological opportunity to be clonal. Or have they evolved traits that confer higher clonality? So what we did is we had 11 populations across our elevation gradient growing in a common garden in our greenhouse. And so we wanted to kind of take them out of their ecological context and measure metrics of clonality. And so we had plants growing for many generations in the greenhouse. And what we did is we looked at the number of runners after a growing season. So this is basically vegetative propagation, runner length, and then the number of plantlets produced per unit runner length. So you could have a meter-long runner and produce 2 plantlets, or a quarter-meter long runner and produce 2 plantlets. You're putting more investment into these clonal propagules per unit runner length on that shorter runner. So what I'm showing here is plantlets per unit runner length on the y-axis and elevation on the x. And what was really cool is that these high elevation populations in a common garden are showing signatures of having this higher clonality. So they're putting more energy into producing clonal propagules. So what we think we have going on is, so what we've shown is that there's a decline in population density on the landscape. These high elevation populations appear to be more recently colonized. I put a soft check because we don't have a properly dated phylogeny. There are declines in effective size, more isolation, and these populations are more clonal. So this is I think it's evidence of increased clonality at a leading range edge. And so we think that range filling likely occurred directionally from low to high elevation following glacial recession. And reproductive assurance via clonality may have been favored during the early stages of colonization. So if these populations at high elevation are highly clonal, are they investing less in sexual reproduction? During 2 seasons, we also measured sexual reproduction in the field. So in these populations in the field, we collected fruit set data and seed set data, and the high elevation populations do have depressed sexual reproduction. And so asexuality at these high elevation limits may be a really poor long-term evolutionary strategy, especially in these stochastic range edge environments. So clonality is great if the clone is really locally adapted and that environment isn't going to change. However, if these high elevation populations experience, severe drought in a given season, some sort of pest in a given season, and you're not recombining through sexual reproduction, that's not great. And so what I wanted to spend the last bit of time talking about is, are these small, potentially clonal populations, do they have reduced mate availability? And do these mate-limited populations exhibit signatures of a breakdown in self-incompatibility? So is sexuality potentially being restored? So I'm going to have to do a deep dive into the self-incompatibility system of this plant, which is common among many plants with self-incompatibility, and it's called gametophytic self-incompatibility. So in plants, many self-incompatibility systems are recognized, but this is the one, and we've confirmed it in this species that this has. So there are what are called S loci, or self-incompatibility loci in plants. And so in this example, the pistil, or the recipient of pollen, is diploid and has S1 and S2. So 2 self-incompatibility alleles in this case. The anther on that same flower is diploid, but it produces haploid pollen grains that can be either S1 or S2. If the pollen S1 and S2 gets onto the pistil of the same flower, there's an sRNase that degrades pollen tube growth. It recognizes like. And pollen will germinate, but it will stop germinating at about the third the length of the style. So let's introduce a new S allele in a population. And so this maternal plant is S1, S3. So if this donor, which is S1, S2, donates both types of pollen grains, S2 can germinate, but S1 cannot. And then finally, if the maternal genotype's S3, S3, both S1 and 2 can germinate. And we confirmed this doing, so Anita did hand crosses, and then we looked at the site of self-recognition using histological approaches to confirm that this has gametophytic self-incompatibility. So self-incompatibility systems can lead to really strong mate limitation, especially in small populations. So this is showing a model of the size of a population and the number of self-incompatibility alleles. So self-incompatibility alleles are maintained at low population size due to negative frequency dependent selection. So if you're a rare S allele, it's really good because you have more potential mates. But we know that as population size increases, S alleles generally increase, and these are 3 different mutation rate parameters, so it always increases. What's really interesting is that our populations of Argentina anserina are exceedingly small in terms of genetic effective population size. They're between 1 and 15 individuals based on the genetic data. And so our populations could be very mate limited due to very few S alleles. However, plants are pretty labile in their mating systems, and self-compatibility evolves from self-incompatibility frequently. And it starts with the what's called leakiness in the self-recognition system. So in this example, we have an S1, S2 pistil, and it's getting S1 and S2 pollen, but the S2 pollen slips by. And so there's might be selection favoring leakiness in the self-recognition in populations that are especially mate limited. This would increase the ability for sexual reproduction uniparentally. So what we wanted to do is estimate mate availability in our focal populations and link it with effective population size. And we did that using ecological genetic crosses. So Anita, pictured here, did tons of crossing to estimate mate availability. We had 15 maternal lines in each of our 13 populations. We hand pollinated, I say the royal we, Anita pollinated these plants with 4 to 6 donors from the same population. And so if a pollination fails, we assume that those two individuals share an S alleles. But if it's successful, they don't necessarily share all their S alleles. So we estimated the percent successful crosses at the dam level and took the mean at the population level to get mate availability. We then characterized leakiness in the self-incompatibility system using self-hand pollinations. So Anita self-pollinated 1 flower per 13 maternal lines per population. It would have been great to do more, but these plants don't make a ton of flowers, and we wanted more flowers for the mate availability. But what we did after the pollinations is collected the styles, and then we stained them and looked at pollen tubes growing down the styles. If a pollen tube from a self-fertilization was successful, that indicates leakiness in the self-recognition. Okay, so I'm going to show data first on mate availability, and it was shockingly beautiful. So this is mate availability, or basically the percent successful crosses in our populations. And this is effective population size genetically determined using RAD-seq data. And then from our crosses, I took the mating tables that Anita created, and we can estimate the number of S alleles in a population from successful versus unsuccessful crosses. What you can see is that as effective population size increases, there's a really dramatic increase in mate availability. And then there's an asymptotic relationship that at about 80% mate availability, it flattens out. What's really interesting is that these clonal populations literally have 1 S allele. So their mate availability is effectively 0, and they're just 1 big clone. So if self-incompatibility is maintained in these really small populations, then sex has been lost. So I'm now going to be presenting data on the leakiness. So leakiness, again, what I'm presenting it is the proportion of pollen tubes that germinated and slipped past that self-recognition system following selfing. And this was kind of beautiful. In our small populations with low effective size, we see quite a bit of leakiness. About 40% of the grains that germinate will slip past that mechanism of self-recognition. And we did this we looked at the tubes that passed 1/3 the length of the style and 2/3, just because where self-recognition happens can be a little bit muddy, but the pattern's generally the same. So these small populations are leaky in their self-recognition. And if we look at that with mate availability, the pattern's really the same. These small populations, highly clonal, low mate availability, but also leaky. So is this leakiness actually increasing sexual reproduction, or are these self seeds actually contributing to populations, or is there so much inbreeding depression that they're not really contributing? I don't have an exact answer to that, but we do know that leakiness is boosting sexual reproduction in the field. So What I'm showing here is mate availability and fruit set in the field on this left panel, and mate availability and seed set in the field. And they're color coded by the degree of leakiness. So blue is more leaky. With increasing mate availability, we have increased fruit set in the field. Beautiful. What's interesting though is these populations that have really low mate availability that shouldn't be producing any fruit are eking out some fruits. And then when we look at seed set, it's pretty amazing. These populations that have no mate availability shouldn't be really setting seed, are having seed set on par with populations that have high mate availability. So leakiness appears to at least be contributing to the production of seeds. So I think whether they contribute to population growth, unclear. So I'm going to revise this kind of sad, dire model I put forth of long distance dispersal of clonal species with self-incompatibility, leading to population decline because of limited recombination. If SI can break down, then recombination could be restored. You get new genotypes. You have the potential for local adaptation. And I'm going to expand. I kind of lied about all of the species being self-incompatible. This is a large species complex, and in New Zealand, all populations are known to be self-compatible. And so this New Zealand kind of all self-compatible is really interesting to me because I think it speaks to the timing of colonization and how long the evolution of the breakdown of self-incompatibility might take. So I think in Colorado, we're seeing really early stages. We're getting leakiness in the SI system. I wouldn't call them self-compatible though. What I was talking about is New Zealand populations being mostly self-compatible. And these populations, I think it would be really cool to kind of date when populations were founded, because if they were founded longer ago, they may have had more time to evolve the breakdown of SI or self-compatibility. And they weren't glaciated, so they potentially could have been more recently colonized. So I think this is a really cool system to look at multiple axes of colonization, like post-glacial south to north, elevational, and island colonization. Okay, so just to recap, high elevation populations exhibit signatures of leading range edges. They're more clonal, have low mate availability, and show the early stages of a breakdown in SI. And leakiness in SI does seem to contribute to sexual reproduction in the field. Whether it's actually leading to more propagules and new individuals is unclear. And I want to say thanks to a number of lab members, the land that we worked on, and Clemson College of Science and NSF for support. And again, thanks to Anita, who did a lot of this work.
Thanks very much for this. It was also very, very interesting. We have some time for some questions. If anyone wants to unmute themselves or raise their hand, feel free.
I have a question, Matt.
Yeah.
I'm really interested in the time scale of these things, like with glaciation. So you have this like gorgeous climb in autonomous self-fertilization ability. And, you know, presumably those populations have been around for, well, I don't know, how long have they been around for? I was assuming like 5,000 years, like the ones we work on, but is that an overestimate?
So the elevation gradient with Argentina, like I said, I need to go back and look at like so I showed that figure of Western glaciation at high elevations. And let me go back real quick.
Let me just ask you my full question, and then I can then you'll know where I'm going. So I guess what I'm wondering is like there's this beautiful gradient that has somehow stuck around. So like once you evolve higher autonomous selfing ability, it doesn't just go away. And yet it like so you keep it. But that's only one of many recent expansions, right? So these expansion processes have like come and like they've happened over and over again. So I guess, yeah, I mean, I guess I was just trying to puzzle through like, why don't you, if it's not bad enough to lose at the expanding front over thousands of years, why don't you just retain it at the kind of the range core, that seed population?
I see. I mean, my first thought would be inbreeding depression could be extreme. And in clonal species, there's also a theory that if if you're making a bunch of clones that can flower, you're increasing more reproductive individuals with your same mating type. And so that leads to tons of gynogamous pollen transfer. And so that, if inbreeding is strong enough, then that should be the main factor. And we don't know anything about inbreeding depression in this system. And that's because in our populations, the leakiness doesn't make many seeds. And so we would have to do like tons and tons of crosses to get the seed produced through selfing to get good estimates of inbreeding depression, but I am up for it and I want to know. But I would say inbreeding depression would be the main thing that would limit just keeping it around. But then again, and like that's why I think having dated populations somehow based on the glacial record and including those New Zealand ones that are self-compatible and looking at inbreeding depression there would be also awesome.
Hi.
So, oh, Louise.
Yes, it's me. Yes. Thank you for the talk. Looks like a very nice place to do some fieldwork.
It's pretty awesome.
I guess it's somehow related, but with this phylogeny that you did, I wonder if you can actually put some dates between the different even like between the population across this elevational range, and actually look at how, because if I understood correctly, this expansion across the latitude, this elevation is through clonal expansion or something. In which case,
yeah, I think, okay, I was looking at, for this talk, I was like, okay, what is the glacial history of this area? Because I haven't, doing this talk really made me start to think more about that. And so there are studies like specifically that date locations of like the timing of glacial recession at specific locations. So I think it's possible to get a better estimate of dating that phylogeny using this like glacial record. And so is that kind of what you're asking about or something different?
No, I was actually wondering whether you can actually look at the speed of expansion through clonal, because like this elevation range that you presented with the different clones, I was wondering whether you can actually kind of have a measure of the speed of expansion through some
So a more genetic
genetic
approach. Yes, I see. What we would need. So I think what we might need to do that is some sort of idea, like an outgroup, so we know what's derived in these populations. because they're all pretty close.
Yeah.
But yeah, or are you suggesting like assigning, like, is an individual clone in one population present in another population? And
Because
that would
yeah,
get at like, did that clone actually do the colonization?
because do you actually observe the same clone across different points in this elevation, or
That is a great question. There are ways to look at that. So we looked at population structure, um, and I can dig back into that, but that's an interesting question. Like, do we see the same clones represented across populations? And I don't have a good answer for you, but I think it's a really good idea. The other thing is like, there are probably so many populations out there that we didn't sample.
yeah, for
So,
sure. Yeah, of course.
like where exactly um, a population, a clone came from, we might not have captured all of it, but it's a good thought.
And just a second question, if I have time, is this leakiness? So I think you tested this leakiness in different population where there's like different estimate of population size. But I was wondering whether actually you tested this how are these population like distributed, and whether this leakiness, is there more leakiness in like this expanding front or
Yes.
not? Yeah.
Yeah,
Okay.
the leakiness tends to be higher in these smaller populations, which are more frequent at the expanding front. We had one population at low elevation that occurs at a wastewater treatment site on a bunch of fill dirt, and it's like one giant clone. And I would argue that that one doesn't probably have a long evolutionary history there, but that one's also pretty leaky. But I think it's because it was just like in fill dirt, a plant got there and it expanded everywhere at the wastewater treatment plant. So that one like
Yeah.
really ruins some of the elevational gradients and some of this. But if we take that one out, yes, then elevation is a really strong predictor of leakiness.
Okay. Super interesting. Thank you.
Greta, do you have a question?
No, it's okay. I realized it wasn't super well thought through. I might follow up with Matthew at some point if it makes sense. But it was super interesting. It's just really got my brain going. Thank you so much.
Thank you all. I'm going to ESEB this summer
Oh, cool.
in Barcelona. So if people are going, I'd love to connect and talk more about this stuff.
I will
Yes.
also be there. Yeah.
Oh,
Anna, you're
cool.
going to. Okay.
I can't find my microphone. Yes, it'll be my first ever ESEB.
Oh, okay.
Oh, wow.
Yeah.
So it would be nice to meet you all in person then.
Yeah, that would be great.
Yeah.
Okay, then if there are no other further questions, then I'd like to thank our speakers, Anna and Matt, again. It was really, really interesting. Yeah, we're very grateful that you chose to speak at our seminar. And I hope to see people again on Thursday, the 3rd of July for our next seminar. So thanks again, and see you then.
Great.
Thank
Yeah,
you.
thank you so much.
Bye.